The topic of today's lecture is saponifiable and non-saponifiable lipids. This picture shows examples of lipids in the human body. As you can see, lipids play a very important role in biological systems because all of the given examples are lipids. For example, vitamin D, vitamin A, vitamin K, bile acids, glycophospholipids, sphingolipids, and cholesterol, eicosanoids, hormones, sex hormones like androgens and estrogens are bright examples of lipids. Now you can see the main functions of lipids. Lipids are an essential part of healthy diet, a major source of energy, an important component of cells and tissues. They also absorb essential fat-soluble vitamins and cell messengers because lipids can be converted into other molecules like prostaglandins, which help cells communicate with one another. They are really good at insulating and regulating our heat. They keep you warm. Here you can see the general families or classes of lipids. Let's describe them in more detail in the next slide. There are two classes of lipids based on ester-containing bond, such as saponifiable and non-saponifiable lipids. Saponifiable lipids have two subclasses. They are simple and complex lipids. Simple saponifiable lipids are waxes and triacylglycerols. Complex saponifiable lipids are phosphoglycerides, glycolipids, sphingolipids. What do these things have in common? It is a kind of linkage. It is an ester linkage. They all have at least one ester linkage. It's a hydrolyzable linkage. It can be saponified. Non-saponifiable lipids are cholesterol, bile acids, steroids, hormones, eicosanoids. They don't have any ester linkage, and that's why they cannot be saponified. It means they are non-hydrolyzable. Let's look at the simple and complex lipids. Simple lipids contain two types of components. For example, an alcohol, one or more than one fatty acids. Complex lipids containing more than two components, such as an alcohol, fatty acids, and plus others. For example, amino alcohol. Fatty acids are long-chain carboxylic acids. They have the carboxyl group at the end, that is the polar region, and a very long hydrocarbon chain, that is non-polar or hydrophobic. Fatty acids have even numbered chains, including the carboxyl carbon. Saturated fatty acids have all single bonds in their structures, and they have straight chain, and usually they are solid at room temperature. Unsaturated fatty acids, they have at least one double bond, that bond non-conjugated. They have kinks and bends in their structures, and usually they are liquid at room temperature. Polyunsaturated fatty acids, for example, linoleic, linolenic, and arachidonic acids, they are named essential fatty acids and belong to group of vitamin F because they don't produce in our body and you should intake them by food. Also, fatty acids can be classified according to the location of double bond since the end of the carbon chain. So we call the methyl end the omega end, and then we count the number of carbons from the methyl end until the first double bond. For instance, linolenic acid named omega-3 because of the position of double bond from the end of carbon chain. There are cis-trans configuration of oleic acid. In a cis configuration, the two functional groups are on the same side of the double bond and the chain naturally blends. A molecule that blends doesn't pack tightly together, and so it is a lot more fluid, whereas saturated fatty acid molecules with elongated conformations pack closely together, and that's why they have high melting points. Linolenic acid is found in plants like flaxseed, walnuts, and soybean oils. Our bodies can convert linoleic acid into arachidonic acid, but in small quantities, and that's why dietary recommendations include foods that have arachidonic acid. Arachidonic acid is found in animal sources like fish, meat, and eggs. Linoleic acid is found in safflower, corn, and soybean oils. Actually, triacylglycerides are lipid esters because they can formed by dehydration of glycerol and 3 fatty acids. Triacylglycerides are non-ionic and non-polar. They serve as energy storage in adipose cells, and they release twice as much energy as carbohydrates. Here you can see an esterification or formation of triacylglycerol. It is a reversible process and a backward reaction named hydrolysis. At the bottom of the slide you can see two different triacylglycerols. The first one named a mixed solid fat. It is mixed because it contains different types of fatty acids. It is a solid fat because the number of unsaturated fatty acids is less than number of saturated fatty acids. In the bottom right we can see a simple liquid oil. It is a simple because it contains same kind of fatty acid, and it is a liquid because all of fatty acids are unsaturated. Hydrolysis of triacylglycerols in our body occur in the presence of lipids. It is a stepwise process, and it begins with the second ester bond. Hydrolysis in a basic medium named saponification because one of the products is a soap. Iodine number indicates the amount of unsaturated fat. Iodine number is the amount of iodine which can be added to 100 grams of fat. The more double bonds in fat, the higher its iodine number. Iodine number of sunflower oil is 130, is greater than others, and that's why it contains the highest number of unsaturated fatty acids than others. Iodination, bromination, hydrogenation are reactions with unsaturated fatty acids. The last reaction is an important industrial process to produce artificial oil. Oxidative processes involving lipids and their structural components are important. Lipid peroxidation is the oxidative degradation of lipids. It is a process in which free radicals steal electrons from the lipids in cell membranes, resulting in cell damage. This process proceeds by a free radical mechanism. Beta oxidation is a metabolic process involving multiple steps by which fatty acid molecules are broken down to produce energy. Unsaturated fatty acids could be tested by bromine water and potassium permanganate solution. Phospholipids are synthesized in the liver and contain glycerol, 2 fatty acids, phosphoric acid, and amino alcohol. Phosphoric acid forms 2 ester bonds, one with the alcohol group of glycerol, the second binds another component amino alcohol. Phosphatidylcholines and phosphatidylethanolamines are bipolar phospholipids. the main components of cell membranes and membranes of cellular organelles. Dipalmitoylphosphatidylcholine is the main component of pulmonary surfactants secreted by epithelial cells of pulmonary alveoli. It protects alveolus against collapsing during exhalation and thereby makes the inhalation of air into alveoli easier. Here you can see bilayer structure of phospholipids. Molecules phospholipids contain two nonpolar tails, formed by two fatty acids, to which one polar head is attached. The phosphate group has a negative charge and a positive charge on the nitrogen at a physiological pH. The polar-nonpolar character of phospholipids is important for stabilization of the phase interface and properties of membranes. At the phase interface, membranes are orientated with the hydrophilic end into the water phase and with the hydrophobic end to the nonpolar phase, for example, air, lipids, or another nonpolar molecules. As I have already noticed, phospholipids contain two fatty acids, one of them saturated, one of them unsaturated. Then phosphoric acid, which forms two ester bonds, one with the alcohol group of glycerol, and the second one with another component, most commonly serine, ethanolamine, choline. The principal structure of glycerophospholipids is phosphatidic acid. 1,2-diacylglycerol, 3-phosphoric acid. That is phosphoric ester of 1,2-diacylglycerol. In the next slide you can see amino alcohols and their metabolic reactions. These amino alcohols, serine, ethanolamine, choline, are interrelated, since ethanolamine and choline can be formed during metabolism from serine by decarboxylation and subsequent methylation. In the body, phospholipids are hydrolyzed in gastrointestinal tract by the enzymes phospholipases, which break off the ester bonds between glycerol and two fatty acids, glycerol and phosphoric acid, and between phosphoric acid and amino alcohol. The reaction in basic medium, named saponification, fatty acids and phosphoric acid salts are formed instead of acids. Steroids are non-hydrolyzable lipids that contain the steroid ring structure, which consists of 4 fused rings, 3 of them 6-membered rings and 1 of them a 5-membered ring. This group includes cholesterol, adrenocortical and sex hormones, and bile salts. The 17th carbon skeleton is made from condensated alicyclic rings called sterene. Rings of the steroid skeleton are indicated with letters A to D. Their carbons are numbered in a specific manner. Cholesterol is the most common animal sterol. We know that cholesterol plays an important role in cardiovascular disease, and I advise to lower our intake of this lipid. Violation of cholesterol metabolism leads to its deposition on the walls of arteries, and as a result, a decrease in the elasticity of blood vessels, named atherosclerosis. In addition, it can accumulate in the form of gallstones. Low-density lipoproteins is actually the bad one because there is more fat in it, and that is not good. High-density lipoproteins is a good one because it has a high amount of protein. Too much fat in LDL, what it does, it sticks on our arteries and causes clogged arteries that could lead to heart attacks, and you can see the blockage forming over time. see in the picture on the bottom right. It is possible if you have high level cholesterol that is high in LDL. Nevertheless, we must keep in mind that cholesterol is critical to many physiological functions. In particular, it serves as a component of membranes and as a precursor for all other animal steroids, including the adrenocorticosteroids, the sex hormones, and the bile salts. In all animal cells, it is a part phospholipid bilayer of cell membranes. Membranes with a larger amount of cholesterol have noticeably lower fluidity and permeability. Cholesterol is a steroid alcohol mainly with a secondary hydroxyl in position 3, often with a double bond between 5th and 6th carbon atoms in ring B. Cholesterol has 27 carbons in the position 17th. It has an 8 carbon branched alkyl. Due to cholesterol's hydroxyl group, it can form esters with fatty acids. Esterified cholesterol predominates in the blood and is stored in small amount in some types of cells that use it as a substrate for the synthesis of other substrates. Cholesterol and its esters are hydrophobic molecules, so they are transported by the blood only as a part of different types of lipoproteins. When irradiated with ultraviolet light, some sterols, for example found in bacteria ergosterol, the ring B breaks off and the formation of anti-rachitic vitamin D group occurs. Vitamin D is found in egg yolk, milk, butter, and fish oil. Ergosterol is a steroid alcohol with a secondary alcohol group in position 3 with two double bonds between 5th and 6th, 7th and 8th carbon atoms in ring B. In the 7th it has a 9 carbon branched alkyl with one double bond. Note that vitamin D3 is obtained not from cholesterol but from its derivative 7-dehydrocholesterol. Deficiency of vitamin D leads to rickets in children and osteoporosis in adults. Bile acids are cholesterol derivatives synthesized in liver. The aliphatic side chain of carbon 17 consists of 5 carbon atoms and includes a carboxyl group. Bile acids are discharged into bile as conjugated bile acids, amide made by binding carboxyl to the amino group of glycine or taurine. For example, cholic acid is converted into glycocholic and taurocholic acid. Conjugation increases the strength of bile acids and therefore their degree of dissociation and solubility. Sodium and potassium salts of these compounds have surface active properties, emulsifying food fats. Bile acids are needed for proper lipid digestion in the intestine. They emulsify fat in small micelles. They also activate pancreatic lipase necessary for the hydrolytic breakdown or hydrolysis of triacylglycerols. Thank you for your attention.